Welcome to Unit 1, Chemistry of Life, Section 1.4, Properties of Biological Macromolecules. Welcome to AP Biology or College Biology. Please note, just as a reminder and a disclaimer, AP Biology is a registered trademark of College Board and is not associated or involved with the development of the products that we're talking about now. Please note that when we're talking about the topic of macromolecules, previously in the previous subsection, you covered macromolecules as an overview. Now, moving forward, you're going to be expected to not only understand the properties of macromolecules, but their structure, functionality, and the nitty-gritty of it. Because of this unit or this subunit or properties of biological macromolecules tends to cover a lot of information, and that's okay because this same information will repeat itself in other sections involved in the same unit. So don't feel overwhelmed if you are in class or in school and are using this side by side. Please note that the information given here can be seen in later parts of the unit, and that's okay. This is just a brief overview, so that way you could piece the understanding of what macromolecules are. So when we talk about macromolecules in general, please note that we're talking about these large molecules that are composed of two or more small polymers combined. So basically we're taking building blocks and we're building them together to formulate four giant macromolecules, which will be the basis of the unit. And those four macromolecules include carbohydrates, proteins, lipids, and nucleic acids. So macromolecules that you're covering are the four major groups. Now let's talk about carbohydrates, which is the first macromolecule that you are expected to know. One of the big things that you need to know is that when you're thinking about sugars, so items like breads or any sort of fruit, they have sugars in them, which means that they have carbohydrates in them. If you're looking for common carbohydrate-related terms, you want to look for the ending OSC. So terms like glucose or fructose or lactose, they all end in OSC, which is a major hint to indicate that we're talking about carbohydrates. Please make note of this because you might in the future have a free response question, and you're expected to understand that these terms are terms associated with the macromolecule carbs or carbohydrates. Carbohydrates have three main elements: carbon, hydrogen, and oxygen, which can be seen in long chains or they can be seen in a ring structure like the ones shown here. So if you see rings, that's your hint. If they have carbon, hydrogen, or oxygen, they're talking about carbohydrates. When we're talking about a very basic or simple carbohydrate, we're talking about something like glucose or fructose, which is a monosaccharide. There is no linkage. There's usually a one-ring structure. It is the basic monomer. And the basic monomer is associated with the term monosaccharide. Mono meaning one. So make note of remembering that. If you have a linked bond between two rings with carbon, hydrogen, and oxygen, you're referring to a disaccharide. Di meaning two, so disaccharide. These are things like sucrose and lactose. The picture on the right here shows a sample of a disaccharide, sucrose, and there's a link right here. That link is called a glycosidic bond. So a glycosidic bond is associated with a carbohydrate, oftentimes in the disaccharide form or in the polysaccharide form, which is a chain of carbs. So having a long chain or a polymer or a poly, meaning many, saccharide is going to be your polymer that has a lot of glycosidic bonds. Now, The function of carbs is associated with energy. If you think you're eating a carb, you're eating it for energy purposes. Structural, like cellulose in plants, and cell recognition, like glycoproteins. Now let's talk about proteins, which is the second macromolecule that you're expected to understand. The way that you think of proteins is you need to understand that amino acids are the building block of proteins. So all major proteins have some sort of amino acid attached to them. Now, there are 20 different amino acids, and so it becomes very hard to identify all 20 of them, and you're not expected to know all 20 of them. What you are expected to know is the basic structure. For example, when you have any sort of protein, you're looking for an amino group. So an amino group is a functional group, and that functional group is associated with an NH2 or an NH3. There is oftentimes a carboxyl group, so that's something like COOOH. So that's something like this part right here. There's usually a hydrogen or a hydroxyl that's usually associated with it, and then some sort of R group. So the R group is something that's arbitrary. It could be small, it could be large, it could be nonpolar, it could be charged. It's just a random group that makes the amino acid very unique. Please note that there are 20 different amino acids, so you're looking for the four major components to really dictate that there is a protein. So look for the structure that has the four, I should say five main elements, which is carbon, hydrogen, oxygen, nitrogen, and sulfur. Or sulfur, I should say. Not always sulfur, but sometimes sulfur. So look for those 4 to 5 main structures, but more specifically, look for the amino group, the carboxyl group, the random hydrogen, and the R group to really dictate what your amino acid is, which would dictate that there's a protein. Now, when we talk about proteins, we talk about primary, secondary, tertiary, and quaternary, and that might be a tad bit confusing. What it really means is what can it formulate into? First of all, if you have a primary structure, you're talking about the very basic coding of an amino acid. For a secondary structure, you're really talking about whether or not it's going to be alpha or beta, which means is it going to be on itself? Is it going to be like on a twist, or is it going to be like flat sheeted? And that's just the secondary structure. So whenever you're thinking of protein, which is secondary, you're saying, is it going to be 3D winded or is it going to be a flat structure? And you're expected to know that. Once you hit tertiary, think 3D. So you're thinking about, okay, now I have my 3D shape. I really have some sort of depth to the protein. And quaternary is a bunch of tertiaries formulating together. That's the largest structure. Now, in terms of linkage of amino acids, they're formulated by peptide bonds. So if you have one amino acid attached to another, that's going to be called a peptide bond. So once again, the monomer is going to be amino acid. That's the most basic structure of a protein. If you have two or more, that's called two, I should say. It's going to be called a dipeptide, which is two amino acids which are bonded together by a peptide bond. And if you have a bunch of them together, that's called polypeptide. So polypeptide is your polymer that is a bunch of amino acids together. Now, in terms of functionality, the most common types of proteins are enzymes. You can also have fibers or storage or transports when it comes to proteins. They have a multitude of different functionalities. Most common is enzyme because you remember that enzymes usually end in ASE, which means that it is a protein. And usually you can think of things that have some sort of change in them, such as pH, ion concentration, and temperature, which would dictate that there is some sort of change in the enzyme, which means essentially we're changing the protein. Now let's talk about lipids. So lipids are a unique macromolecule because they're not really considered a polymer. The thing is, lipids are unique because they're water-fearing. I want you to think about this test tube over here, which we have oil and water. The thing is, an example of a lipid is an oil. When you add oil to water, they tend to separate, and that's because the lipid oil is water-fearing or hydrophobic. So they have a fear of water and they're nonpolar. So what you need to remember is that typical lipids, which include things like fats and oils, think like butter, and phospholipids and steroids like cholesterol, they're going to be hydrophobic, which means that they're water fearing, which is interesting because they're not like other macromolecules. They commonly hold the structure with four main elements: carbon, hydrogen, oxygen, and sometimes phosphorus. So something like phospholipids would have phosphorus in them. Not always, but that's something to note. They are solid at room temperature. I want you to think of something like butter. Butter is a type of lipid. It is going to be solid at room temperature, which leads to the next type. So they're separated into two main categories: saturated versus unsaturated. The thing is, the carbon-carbon bonds are going to be single. There's only one link between them, and that's what makes them saturated. So they're going to be solid at room temperature. So think of, like we said before, butter. And unsaturated would have the double-double bond. So think of liquid at room temperature, so something like oil. And the functions are commonly energy, insulation, and cell membrane structure. When we're talking about the topic of nucleotides, we're talking about the understanding that the building block or the monomers are nucleotides, which basically are our five nitrogenous bases: adenine or A, thymine or T, uracil or U, guanine or G, cytosine or C. It's important to note that when we're talking about nucleotides, we're talking about the understanding that the nucleic acids are in two different forms. And those two different forms are DNA or RNA. Those are our polymers. And the unique thing about nucleic acids is we're talking about the functionality of genetic information. So when it comes to DNA, we have four main nitrogenous bases: A, T, C, and G. The term D means deoxyribose, and that DNA is also double-stranded, whereas RNA is going to be single-stranded, sugar associated as ribose, and we do have four similar nitrogenous bases, but instead of thymine, you utilize uracil. The bonds between the nitrogenous bases is going to be hydrogen. So you have a double bond between A and T or A and U, and a triple bond between C and G. And there's going to be hydrogen bonds. On the backbone, or the linkage between the different nitrogenous bases that make up DNA and RNA, the linking between the two, or the many, to form the polymer of DNA and RNA, is going to be the phosphodiester bond. So don't forget the difference between the bonds, because this is a common misconception. Between the nitrogenous bases, you have hydrogen bonding. Between the idea of having a polymer, or many nucleotides, you're going to have a phosphodiester bond. Please note that the elements associated with the nucleotides are listed as CHO, N, or P, and P. So it's important to note that the difference between the two. So as a refresher, you have nucleotides. The main types of nucleotides are RNA and DNA, RNA being single-stranded, DNA being double-stranded. They have the same nitrogenous bases, with the exception of thymine for DNA and uracil for RNA. The bonds between the nucleotides are going to be hydrogen, meaning between A and T is going to be your double bonds, and between C and G, which are bonded together, are going to be your triple bonds of hydrogen. But the backbone is called the phosphodiester bond. Please also note that A bonds to T and C bonds with G, or A will bond with U for RNA. Now, the idea is that the nucleotides of A and G are called purines, and that C and T are called pyrimidines. So you have one purine that's always attached to one pyrimidine. Now for, of course, RNA, the T would be U. So that's something to note as well. So if you're talking about this main type of nucleic acid, we're talking about, remember, the functionality of genetic information. We're talking about DNA and RNA, and we're talking about the differences between the two. So now that we've reviewed all four major nucleotides, this table or summary talks about each of the macromolecules, each of their structure, the type of linkage, the types, functionalities, and examples. If you want to take a screenshot of this, this will be helpful as well. I hope that you enjoyed this debriefing of macromolecules and it helps you really understand breakdown and differences between the four major groups.
Hey, you want to know a little secret? If you hit the subscribe button, you'll be able to get up-to-date information related to AP Biology and the digital SAT math test. Making sure that you're able to understand the content is just the first step. So if you want updated additional information, make sure to click the link tree in our bio. We hope that you subscribe because each new subscriber gives us motivation to create more up-to-date content for you, students, parents, and teachers. Enjoy!